Hello students, so you all know that our unit plant physiology is going on and chapter is photosynthesis in higher plants. Today we'll study about C4 pathway and H and SLEC pathway. So as I have already told you, C4 pathway, that is first carbon fixation product is oxaloacetic acid, that is having 4 carbon molecule. So plants that are adapted to dry tropical regions have the C4 pathway. C4 oxaloacetic acid is the first carbon dioxide fixation product. The plants with C4 pathway does follow Calvin cycle as the main biosynthetic pathway. C4 plants differ from C3 plants by having special type of leaf anatomy. They tolerate high temperatures, they show a response to high light intensities, they lack a process called photorespiration and have greater productivity of biomass. It means they can survive in a more stressful environment. Anatomy of C4 plants differ from C3 by having large cells around the vascular bundles, which are called bundle sheath cells. And the leaves which have such anatomy are said to have Cran's anatomy. It's like vascular bundles के चारों तरफ जैसे गोल बुके होता है, wreath of flowers is like that Cran's anatomy. The bundle sheath cells may form several layers around the vascular bundles and have large number of chloroplasts, thick walls impervious to gaseous exchange and no intercellular spaces. The pathway followed by C4 plants are called H and SLEC pathway is again a cyclic process. The tissues involved in the process of C4 pathway are mesophyll and bundle sheath cells. The process starts in mesophyll cells where carbon dioxide is accepted by primary carbon dioxide acceptor, which is 3 carbon molecule, and that is called phosphoenol pyruvate, catalyzed by PEP carboxylase enzyme. So mesophyll cells lack Rubisco enzyme. The C4 acid, oxaloacetic acid is formed in the mesophyll cells, which then forms other 4 carbon compounds like malic acid or aspartic acid in the mesophyll cells. Malic acid formed are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release carbon dioxide and 3 carbon molecule. The 3 carbon molecule is transported back to the mesophyll where it is converted to phosphoenol pyruvate again, thus completing the cycle. The carbon dioxide released in the bundle sheath cells enters the C3 or Kelvin pathway, a pathway common for all plants. Uh, the bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase oxygenase, commonly called Rubisco, but it lack phosphoenol pyruvate carboxylase. Uh, Thus, the basic pathway that results in the formation of the sugars, the Kelvin pathway, is common to the C3 and C4 plants. Here you can see in the H and SLEC pathway, mesophyll cells and bundle sheath cells are present. And in between plasmodesmatas are present from which substances from mesophyll cells can go to bundle sheath cells. Now here you have to note that mesophyll cells lack Rubisco enzyme. The C4 acid oxaloacetic acid is formed in the mesophyll cells and bundle sheath cells are having Rubisco but they lack phosphoenol pyruvate carboxylase enzyme. So this is the diagrammatic representation which I have already discussed with you in the earlier slides.